Welcome back to the second session for ECN. It's great to see a lot of people in person this year, but also I'd like to give a big shout out to the approximately 40 virtual participants that are online right now. It's really great to be able to host this hybrid format for the Entomological Collections Network annual conference. So this year, it represents the third symposium of the Entomological Collections of the World Tours video. It was a fantastic success during the virtual conferences that we hosted. And this is our first attempt doing this, both live in person and online. So when we first started this, we've so far hosted about 30 different entomological collections tours so far. The first year spanning 14 countries. The second year, we did 6 countries and 8 tours. And this year, we are fortunate enough to have another 12 different collections tours from 7 countries around the world. So this symposium is going to be moderated by myself, Nicole, Gunter, here in person. And then online, we also have Jennifer Giron, who will be co-moderating with me. And we have her here to help translate many of the question and answer sessions that we have online. So a lot of these videos are recorded in different languages with subtitles. So this is a really great opportunity to showcase collections around the world in their own language. So this format, each presenter has 15 minutes allocated to them. The format may be either a live in-person PowerPoint presentation, traditional style, or a pre-recorded video tour of their collection. We will also have the live question and answer a time available. At that time, if the person is here, the presenter is here in the audience, they can come up on stage to answer the questions. So with that, we will take it away with the first collection tour of the Florida State Arthropod Collection.
Maybe. No. Sorry.
Oh, hi. Oh, being here, you're either lost, confused, or on a quest. I'm Paul Skelly, head curator of the Florida State Collection of Arthropods, which is where you found yourself. This collection is part of the Florida Department of Agriculture and Consumer Services Division of Plant Industry, where our primary mission is to protect the plants in Florida by diagnosing plant pests. Anything that's suspected to be a pest can be sent in, and we'll help figure out what it is to know how to best respond to the find. The collection, which started as a reference collection within our organization, started in 1915, and in the 1960s was merged with other collections within the state to become the Florida State Collection of Arthropods by Florida statute. At that point, we had developed a research associate program so that we could collaborate with international experts, national expert students around the world. A few years later, the Center for Systematic Entomology was established, which is a nonprofit support group that offers travel grants to students to come visit the collection. Since that time, the collection has grown tremendously in numbers. We have over 10 million pin specimens, 200,000 microscope slides, 65,000 vials of insects and other arthropods, thousands of bulk samples that still need to be processed, and a small but growing fossil insect collection. With the growth of the collection came the growth in staff. We have 8 PhD curators and over 16 additional staff that do our day-to-day business here. So why don't we hear from some of them about different aspects of the collection.
Hi, I'm James Hayden, curator of the Lepidoptera. The Lepidoptera holdings of the FSCA are curated in the McGuire Center of the University of Florida in a building next door to the DPI campus. Combined, the collections comprise several million specimens, one of the largest in the world. Regarding moths, our strengths are in Florida and the southeastern United States. Many accessions started in the 1960s with Howard Weems and company, and things really accelerated in the 1970s. Surveys in Florida are ongoing, and new state records should be deposited in the FSCA. Notable donations to the center include the moths of Dickel, Cavell, Belmont, and many amateur collectors in Florida. We also have the collections of Heitzman, Sullivan, Brew, Leuschner's Noctuids, Heichberger material from Europe and Africa, and the Kawahara lab globally. Tropical moths are mainly the efforts of Hepner in the Neotropics and Southeast Asia, also in the Caribbean by Woodruff and Dow, and in Malawi by Murphy. Butterflies include the Allen Museum, the collections of the Emil brothers, and many others. We also have a larval collection of an estimated 30,000 vials.
Hi, I'm Gareth Powell, curator of Coleoptera and Odonata here at the FSCA. In total, we have about 2.1 million curated beetles representing over 78,000 species. We are currently working to publish a catalog of our type specimens, which include around 900 primary types across the order. Our holdings continue to grow at an impressive rate with more than 50,000 beetles added on average per year. The collection is especially strong for Florida taxa, but we maintain an impressive number of specimens from across the US and Neotropics, with particularly valuable holdings from the West Indies. Due to past staff or relationships built through our research associate program, we have a great representation within the scarabs, buprestids, and meloids. But with over 40 years of continuous curator expertise in little brown beetle groups, it is not surprising that several of these families are among the most diverse and important in the hemisphere. Specifically, we have Strohecker's endomycids, Mike Thomas's years of building the sylvanids, cucugids, and laemaphiliids, and Paul Skelly's work with irritilids, a commitment I hope to continue within the nitidulids. Lastly, with the addition of both the Giesbert and Wappus cerambycic collections, our longhorn beetle holdings are among the best and most comprehensive in the New World. I also oversee the Odonate collection, which currently stands at around 1 million specimens. For the last 25 years, this collection has been largely maintained by Bill Moffrey. During this time, several large and important collections have been donated and incorporated. Most notably, the private collections of Carl Cook, Thomas Donnelly, Sid Dunkel, Dennis Paulson, and Ken Tennyson, just to name a few. With more than 3,700 species, this is possibly the largest and most complete collection of dragonflies and damselflies in the world.
Welcome to the sections of Hymenoptera, Neuroptera, and Mollusca at the Florida State Collection of Arthropods. Our collection of Neuroptera was developed by Lionel Stange, a world expert on antlions, who worked in the collection until his passing in 2020. Lionel collected throughout the world, and as a result, our holdings of this taxon are cosmopolitan and are estimated at 36,000 specimens. The Hymenoptera collection is the largest in the southeastern United States, with approximately 850,000 specimens. It contains significant holdings of ichneumoniid and braconid wasps, including many type specimens. As a result of efforts by Virendra Gupta, this collection has a strong emphasis on material from the Indian subcontinent. Our collection of neotropical orchid bees is one of the largest in the world, and our bee collection is currently the focus of an NSF-funded digitization project. We are in the process of making specimen data and high-resolution images available to the public and the scientific community to facilitate studies on pollination biology and how species distributions change in response to global warming. Taxonomic research in the collection currently focuses on parasitoids in the family Platygastroidea, which contains numerous biological control agents. Specifically, we are studying parasitoids of invasive stink bugs and pestiferous gall midges. This contributes to the preservation of Florida's agricultural economy and simultaneously expands the breadth of our Hymenoptera collection.
Hello, my name is Dr. Erin Powell. I'm the curator of the Coxoidea and Alarotidae here at the FSCA. Dr. Susan Halbert serves as the curator of the other Hemiptera. In total, the Hemiptera collection has about 450,000 pen specimens, over 75,000 slides, and several thousand vials of material and alcohol. Hemiptera is one of the most economically important groups as major pests of agriculture and vectors of plant pathogens. In addition to its vast academic value, the FSCA serves as an important reference collection to protect Florida's agriculture. Dr. Frank Mead was the original curator of the Hemiptera, excluding the Coxoidea and Alarotidae. He specialized in Saxiidae, so this part of the collection is diverse. Since 1995, Dr. Susan Halbert has curated this part of the Hemiptera. She has specialized on the vectors of plant pathogens, namely the Florida psyllids and aphids, so these parts of the collection are well curated with good species representation. We also have several notable donations from research associates. Archie Tissett and Clyde Smith contributed to the aphids, Joseph Eager, Pentatomids, Alan Sanborn, Cicadas, Paul Freytag, Leafhoppers, Richard Baranowski, Ligioidia, Mark Rothschild, Membracity, and Lois O'Brien, Fulgoroidia. The Coxoidia and Alarotidae collection is mostly made up of slide-mounted material, which represents about 1,000 species. This collection is one of the largest and most diverse in the country, with only the USNM, CDFA, and UC Davis having more holdings. Several curators have contributed to the collection, including Edward Berger, C.E. Wilson, George Merrill, Wally Deacle, Avis Hammond, Greg Hodges, Ian Stocks, and Z. Ahmed. Doug Miller has donated many specimens as well as several paratypes.
Hello, my name is Felipe Soto. I am curator of Thysanoptera, Colembola, and non-insect arthropods, and I oversee the alcohol and slide collection. The alcohol collection comprises some 200,000 vials and 1.1 million individuals, 46% of which are identified to species or genus. The slide collection includes 9,000 slide boxes and 637,000 slides. Many slides are vouchers of ecological or faunistic surveys done between 1910 and 1940 and represent the only record of some soil microarthropods for areas of South Florida that are now completely paved over. The alcohol collection is particularly rich in arachnids, and although the bulk of the vials are spiders, there are also important holdings of scorpions, datiral legs, pseudoscorpions, and sun spiders. The most important alcohol historical collections are mumas, sun spiders, causis, myriapods, and much more pseudoscorpions. The FSCA also holds the important historical type collections of pseudoscorpions, mites, and thrips by much more, Denmark, and Watson. Other significant slide collections include fleas, lice, hemipterans, and sandflies.
Hello, I am Eric Rodriguez, curator of Dictera and Minors orders at the FSCA, and I am excited to share some of our holdings with you. With over 900,000 specimens, the FSCA is one of the major North American fly collections. We have about 2,600 drawers with pink specimens, at least 135,000 slide mounted specimens, 185 primary types, and many specimens to be processed. Of the 162 currently recognized families of Dictera, 113 families are represented. Due to past worker house at the FSCA, relationships built through our research associate program, taxonomically, we have great representation within the family Acilidae, Tabanidae, Syrphidae, and Ceratopogonidae. The main sources of this family have been from martin, Virtual, wings, plankton, and worms. Lastly, the collection of fruit fly larvae is possibly the largest and most complete collection in the world.
Hi, I'm Kyle Schnepp, Collections Manager here at the FSCA, and we hope you enjoyed this brief tour. We have much more to offer as our collection is continually growing. If you'd like to plan a visit, have any questions, or request a loan, you can contact us at fsca.fdax.gov. Additional information can be found at our new website, thefsca.org. Several of us will be here at the meetings and can answer any questions you have.
Those are Any questions? And there are other people here that have been at the FSCA a lot longer than me that probably answer them better as well. That's a great question. And if anyone told me, we would order some more because they don't last very long and a bunch of them are broken. So if you're thinking that's a solution, that is not a solution. Yeah.
We have designs for those. You can make that a wooden acrylic that we have in production just like this. There was a similar instructional handout for the grant newsletters for making those plans for the final rack.
Anyone else? Thank you very much.
Thank you.
Excellent. Well, thank you very much for our first collections tour. So next up, we have a tour of the entomological collection
Hello, my name is Dimitri Forero.
of the Instituto de Ciencias Naturales from Bogota, Colombia. And Dimitri Forero is joining us online. So once we played the video, we have the opportunity to tune in for those question and answers.
And I'm an assistant professor of the Universidad Nacional de Colombia. I work at the Instituto de Ciencias Naturales, and together with Carlos Sarmiento, we would like to present our entomological collection. The Instituto de Ciencias Naturales has a long tradition about documenting the biodiversity of Colombia. We are composed mostly of botanists and zoologists, and the entomological collection specifically started in 1928 with the efforts of Luis Maria Murillo Quinche. He was an agricultural entomologist, and he amassed an important insect collection related to agricultural problems. Unfortunately, most of his specimens have been lost through time because of moving across institutions and with a lack of careful curatorial practices for his material. The Instituto de Ciencias Naturales was formally founded the way we have it today in the 30s, and in 1939 it was hired a German entomologist called Leopold Wittker, who collected widely across Colombia and was focusing specifically on membrasids. Later on, in the 70s, we have the fortune to hire two more entomologists. One, Isabel Zanabria de Arevalo, was working on several dipteran groups, and another entomologist who was working again with membrasids. Later in the 90s, we have 4 more entomologists joining the team, and more recently, I have joined the Instituto de Ciencias Naturales in the entomological section. The entomological collection is very important, not only because it's comparatively with other Colombian entomological collections, it's an older collection, but also because we have collections from all over the place in Colombia. And if you look through time, we have specimens collected in areas in which probably when you go back, there are no natural areas extant. So this is very, very important to document the changes through time across Colombia. The collection is housed in compactors that help us to save a lot of space. And for the pin dry collection, we have about 2,000, more than 2,000 Cornell type drawers that house about more than 150,000 specimens. Similarly, for the ethanol collection, we have all the vials stored in compactors. We estimate that we have more than 100,000 10 milliliter vials, and these vials are with 75 through 95% ethanol. In addition to the pin dry collection and the ethanol collection, we have about 750 small wooden drawers. These wooden drawers are meant for material that is through the course of curatorial processes. We have either a few ones that have glass lids and the other ones that are completely wooden boxes, and these help us to prepare material before entering in the main collection. Of course, because it's an old collection, we have a lot of material that has not yet been curated adequately. We still have several specimens housed or placed in paper envelopes. Through the efforts of collecting past curators and through the research of several researchers, a lot of the diversity of entomological diversity of Colombia is housed in our institution. We have about 250 holotypes, and the collection is mostly based on the efforts of teaching efforts with students. It's very important, the amount of material that is collected every year with students. Every semester, we have at least a 2-week field trip in different parts of Colombia. We try to go to places that have not been sampled before. We use a lot of different insect collecting techniques, trying to improve every time the type of collecting efforts that we have with students. Not only the students make a lot of effort trying to sort, prepare, and adequately collect the specimens that represent a good addition to the collections of our institution, but also, and more importantly, we are preparing the new generations of entomologists that are superbly capable of collecting insects and preparing them for future studies. These specimens, of course, are going to impact positively in the research that we do. Depending on the interest of each of the curators through time and the actual curators, we have different strengths in our collection. So I would like to present you the groups that historically have been representative of the ECN collection. These specimens, of course, are being used not only by graduate students of our institution, but other institutions in Colombia, and also for visiting researchers nationally and internationally. In this case, we have the director of the Berlin Museum, Dr. Christoph Hauser, looking at very happily through some of the butterfly collection here. And we have one of the best butterfly collections in Colombia, thanks to Gonzalo Andrade, who has been taking care of this collection for a long time. Or another Lepidoptera group that has been very well curated is Saturnidae, through the efforts of Ángel Amarillo, that keeps looking into this interesting group. Another group, Holomechabolus group, that is important in our collection are the Diptera in two groups mainly. The Simulidae that was curated by Paulina Munoz is a reference collection nationally, and we have another important collection of the Agromycidae that was curated by Isabel Zanabria de Arevalo. And not only we have pin dry specimens, but we have some other Diptera that are in glass lights. And for the Hymenoptera, we have several imps groups that have been focused on curatorial efforts. One of the first is the Formicidae, thanks to Fernando Fernandez. This is probably one of the 3 best collections of ants in Colombia that has a very good representation across the country. The other group of Hymenoptera that is very well represented is the Braconidae and Ichneumonidae as a whole, thanks to Carlos Sarmiento again. And also we have a very good collection of Vespidae, again, thanks to Carlos Sarmiento. Both Fernando Fernandez and Carlos Sarmiento have helped build a very good Hymenoptera collection as a whole, ranging from, for example, very small Chalcidoids through really large Pompilids. So everything in between has been collected, curated, and it's always open to researchers that would like to have a look into Hymenoptera groups. In addition to pin dry specimens and the alcohol collection, we have a fine histology collection for Vespidae, particularly because Carlos Sarmiento is interested in several aspects of the fine morphology of these groups. Thanks to the curatorial efforts of late Germán Amat, we have a good collection of cassowlet beetles and also of other scarabaeoid groups of Coleoptera, and we have as well some other beetle groups, probably not as well curated as we would like to, but it again is open to other researchers. Moving from Holomechabolus groups, the Hemiptera, specifically the Mebracid collection, is one of the best in Colombia. Again, thanks to the collecting efforts of Leopold Bidker, and more recently, since I joined the ECN, I would like to build upon the good collection efforts of many, many years and many students and researchers to try to have a very, very good collection of Heteroptera through time. So one of the challenges that we have right now, one of the most important ones is how to better preserve the specimens, thinking in how do we access DNA and how do we do very good morphological analysis with the limited resources that we have available. Of course, something that we would like to have every time is an improved workflow from the field to the collection. How do we integrate all these efforts seamless to impact positively the collection? So this is the brief presentation that I would like to have, and I will be more than happy to answer any questions that you might have. Thank you very much.
So we have Dimitri joining us online. For those of us who are in the room, if you can ask your question, then I will repeat it into the microphone so he can hear us. So does anyone have any questions here or also online? I personally think it was fantastic to be able to see a collection that I never would have before. In terms of visiting Colombia, is it difficult as a foreigner to get collections permits and to collaborate with people? And what is the process involved? Do we have Dimitri? Dimitri, I think you're, we cannot hear you on this side. It may be on our audio though.
I think it's not coming through. Yeah, we can't hear him. It's not picking up.
Yeah, we can see that your microphone is on, but we can't hear it on this side. Is anyone else able to, who is online, able to test their microphone?
Hello, can you hear me?
Yes, Jennifer.
Yeah, so Jennifer, we can hear you.
Dimitri, can you say no? Is your microphone plugged?
Well, we might, if you could respond in the chat, that would be very useful. But thank you so much for joining us virtually. It really was fascinating to see the ECN. And it's very unfortunate that we couldn't hear that response. So the next tour we have is of the entomological collections of the Bean Life Sciences Museum at BYU in Provo, Utah. And this is presented by Tegan Mulford, Rebecca Page, Anna Monson, and Shawn Clarke.
Hello, and welcome to the Bean Life Science Museum. Let's head inside.
Inside the museum, we have a stunning exhibit area with a particular focus on stewardship and conservation. It is utilized by many families and schools in the area, as well as undergraduates at BYU.
We showcase live animals, as well as many interactive displays to facilitate learning. Now let's go to the best part of the museum and the reason we're here today, the invertebrate collection. The Bean Museum houses about 3 million curated insects. We are classified as a Tier 2 collection by Cobb et al. In 2019, making us the 32nd largest out of the 233 collections they surveyed in North America. We have integrated several historical collections, including Vasco M. Tanner's original collection and the Tidwell Butterfly collection. You can even see a specimen from Vasco M. Tanner right here. Tanner became chair of the Zoology and Entomology Department in 1925. Through his work between the years of 1951 and 1971, the collection grew from around 300,000 insects to over 650,000 insects. It's because of him that we have the foundation for our collection today. And now the BYU Tanner Entomology Club is named in his honor. The collection wouldn't move into the physical space of the Bean Museum until the 1970s, but over time it's grown to take up more and more of this top floor. We share this floor with the botany collection, which has about 500,000 of its own specimens. The Bean Museum also has an important lichen, bird, herpetology, and mammal collection. The invertebrate collection is also a place where many undergraduate students, graduate students, museum staff, and volunteers work. Dr. Michael Whiting is the current curator, with Dr. Riley Nelson and Dr. Seth Bybee as assistant curators. Dr. Shawn Clarke is the collections manager, with Dr. Richard Bauman still close by as emeritus curator. They work with 3 research affiliates, a small army of 16 undergraduate lab assistants, and several graduate students. I'm an undergraduate lab assistant here, and we get a chance to work with insects in vials, envelopes, and slides, as well as on pins. I know I've been grateful for the introduction I've gotten to identification on the job by watching Dr. Clarke and others sort, as well as having access to keys and other resources for my own specimens. We've even gone on small collecting excursions to make sure we understand the basics of collection. But don't take my word for it, here are some other lab assistants.
I am Amy Dixon, and I am a sophomore here at BYU in the Biodiversity and Conservation program. Working with Dr. Clarke has been really cool. I've learned so much about insect diversity, and I discovered my passion for insects that I think I might not have noticed otherwise.
Okay, my name is Cole Moulton. I am a senior at BYU and I'm studying biodiversity and conservation. I think there's 2 things I really enjoy about working at the museum. First is meeting a lot of other people who are really interested in entomology or just biodiversity, and getting to see the great collection, everything that we have here, and to look at it together. But the second best thing is that I've actually had the chance to receive something called a Cura Award. It's something BYU offers. It's a college undergraduate research award. And so I wrote an application stating that we had an unidentified species of leaf beetle here, and I really wanted to identify it and write a description, and that BYU was gracious enough to give me a grant to do so. And so it was a great experience.
The students here can learn to pin and curate insects, database them and scan, and take high-resolution photography. Nearly 200,000 individuals of our pinned collection are digitized, with 95% of that number georeferenced, thanks to funding from an NSF grant in 2014. Because of this effort, these digitized specimens can be located on the Symbiota Collections of Arthropods Network, or SCAN, facilitating research with these important historical specimens. Now let's say hello to Dr. Shawn Clarke, our collections manager.
I am Shawn Clarke. I am the collections manager at the collection at BYU. Most of the curators at BYU have been coleopterists, and so needless to say, that is a strength of the collection. But also we have an emphasis on aquatic insect groups, especially on Plecoptera. Right now with NSF funding, we are databasing the flea collection at BYU, which is absolutely outstanding, mostly due to the activities of adjunct curator Michael Hastreiter. We have certainly one of the largest flea collections in the world, among the largest, not only in size, but also in taxonomic representation. There are few collections in the world that have more species represented than we have at BYU. We encourage loans at BYU. Anyone desiring specimens from BYU should simply send a request to the curators or collection manager here at BYU. We're happy to loan out specimens. Generally, we will not loan specimens to private collectors or private individuals. The specimens are loaned to faculty members, or in most instances to the university where the faculty member is on staff. With that in mind, individuals are certainly welcome to borrow specimens from the collection also, as long as they have some sort of a institutional affiliation. The best insect in the collection is any chrysomelid beetle that happens to be in the collection.
The Bean Museum doesn't just have pinned specimens. About a quarter of the collection is frozen specimens in -80 degrees Celsius for next-generation sequencing and other high-output data. These specimens are carefully sorted, and they've been used in many projects. This is where a lot of specimens from recent collecting trips are stored. For example, many specimens from recent trips to Vanuatu, Rwanda, and Vietnam are stored here. The representatives from key species are sometimes pinned to aid in identification. This portion of the collection is used constantly by BYU's labs. So we've talked about pinned and frozen specimens, but what about aquatic insects? For this next part, you'll have to follow me to the basement. Since the ethanol is such a fire hazard, this room is on a closed circuit, so let me just turn everything on. The Bee Museum also houses nearly 1 million aquatic insects in the Bauman Aquatics Collection, named after Richard W. Bauman in 2009. Each specimen is carefully curated and sorted so that they're easy to find. We're now moving some of them into new vials to prevent drying out. Since there's so much ethanol in this room, in order to keep ourselves and the specimens safe, this room was very carefully designed and constructed to prevent fire. Thanks for visiting the Bee Museum. We're excited to see you soon.
We have a couple of minutes for questions. If you could just repeat them so that people can hear them.
Perfect. I'm Tegan. Hi.
So, Shawn, what's the background with having a large series of specimens from Rwanda? When were they collected?
Is Dr. Bybee, are you in here?
They were collected in 2012 and 13.
Okay, but under what project or agreement that happened?
So it was just a directed trip in collaboration with the government there, focusing mostly in the two large national parks. And the collections are mostly focused in odonates and Thailand.
He was also in Vanuatu and Vietnam.
What's that?
You were also.
The New Boy Forest?
Yes.
And which other?
It's Akadera.
Yeah. I'm from the genocide area.
Okay.
Yeah.
Well, let's catch up later.
I was there during the genocide.
All right. Wow.
I have a question about all of the student involvement. How do they find their way to the collection? Is it active recruiting done by the Bean Life Science or is it through Clarke?
Yeah, so I actually worked as an undergraduate in the Bean as well. And it was, I worked with a professor who told me there was an opportunity to database, and the collection's kind of like, if you're on campus, it's like on a hill, it's like the highest point. And so it's, you know, a short walk from like student housing. So it's also, everyone knows that it's kind of like the best job you can get, pretty flexible hours. I went to one of the Vanuatu trips. They've now gone twice. And we mainly focused on nitidulids and then odonates. And I was on the naiad team and then also lampierids. And then some general, like any cool, you know, if we come across like a cool phasmid. Yeah. Yeah.
Thank you very much. So in order for us to keep to the schedule, just in case someone is joining us online at the exact period of time, we are going to have a 5-minute break right now. Like, I feel we caught up.
Yeah.
Must be. Excellent. If everyone can slowly make their way back to the seats so we can stay on the schedule.
They didn't listen.
Didn't listen. Teachers clap. Got a hold of.
Thanks, Mike.
So next up, we have a tour of the Canadian National Collection of Insects, Arachnids, and Nematodes. Owen's not here to join us today, Owen Lonsdale, but he has pre-recorded this video for us. and the collection is in Ottawa in Canada.
Hello everyone, my name's Owen Lonsdale. That's me in the stock photo above. And I couldn't be there with you today, but I'm happy to have been invited to send this presentation to you discussing my collection, the CNC, which is the Canadian National Collection of Insects, Arachnids, and Nematodes. So we're located in Canada's capital, and we're on the Central Experimental Farm, which has been doing agricultural research continuously for about 140 years now. And we're in the Neatby building, and we take up most of the 3rd and 4th floors on the south wing. We originated in 1886 under the first Dominion entomologist James Fletcher, and the agricultural library we have here is over a century old, and that originated under our second Dominion entomologist. We had a maximum of 49 scientists in the 70s, which I'll discuss later, but we have a very good concentration of researchers right now. We have 16 scientists, 1 biologist who's the collection manager, which is me, and we also have 18 technicians and 1 artist. We've been under the care of Agriculture and Agri-Food Canada since 1917, so we have a strong agricultural mandate. So we protect Canadian bioresources, we monitor for invasives, pests, and beneficials. We facilitate trade in the economy, we support our partner agencies in government, in academia, in industry, the public, and a lot of others. Because we've got this great reference collection and a large concentration of taxonomic expertise, we've been designated National Diagnostic Center. So we have a regulatory agency here in part, CFIA. We have the Canadian Forestry Service here, and I run the National Identification Service as well. And in some years we get upwards of 10,000 specimens. So that keeps us quite busy because we only have about 20 people here who can do these identifications. Our primary mandate, however, is research. And while we do a broad spectrum of different types of research, most of this can be categorized as systematics. And every year we describe at least 40 peer-reviewed studies in journals and book chapters, etc. And we describe at least 120 to 150 new species each year. But in some years we have 1 or more individuals that describe at least that many themselves. We also facilitate science. So at least in pre-COVID years, we hosted at least 50 visitors, and we would send out 100 loans every year that included sometimes up to 25,000 specimens. With regards to our holdings, we have about 18 million specimens, which makes us the largest repository of biodiversity in Canada and one of the largest collections in the world. Canadian specimens make up about half of our collection, but we do have representation from virtually every country in the world except for a few smaller countries, some of which are city-states. We add about 50,000 new specimens each year, so that means about every 20 years we're going to be adding 1 million specimens. We've recently completed a taxonomic inventory of the collection, and we have in the collection for described species, of course we have many more undescribed, we have 150,634 described taxa. And Coleoptera is by far the most diverse for named species in our collection, followed by Diptera, Hymenoptera, and Lepidoptera. And for name-bearing types, we have almost 18,000 name-bearing type specimens. And for these, Coleoptera is 3rd place, and in 1st and 2nd is Diptera and Hymenoptera. When you look at the top collectors for the collection, you can probably recognize every one of these names because you've probably used their material in your own work. Our top collector by far seems to have been Dick Vakaroff, who recently passed away, unfortunately. And he's probably collected almost 500,000 specimens that are deposited in the CNC, so he was incredibly prolific. Now, this is a graph of number of specimens collected per year in the CNC. And at the beginning, you can see our oldest specimen in 1809, which is strangely enough from the Canary Islands. Our oldest Canadian specimen is that butterfly in the top corner, which was collected in 1846. And in the middle of the graph here, you can see a large spike. And this might lead you to ask, what happened in this period, around the 40s and 50s, that made the government develop an interest in growing a collection of invertebrates? An entomologist from British Columbia, G.J. Spencer, noted that this interest owed an enormous and hitherto completely unrealized and unacknowledged debt to Japan and to the Soviet Union, which I will explain. North America, you can see, is on the right. Japan and Russia are on the left. And during the Second World War and during the Cold War, we were quite terrified that we were going to be invaded in the Nearctic from across the Pacific through the Bering Straits, or through a land invasion across the Bering Straits, or perhaps from Russia, at the time Soviet Union, sending missiles over to blow us all up. It seems a little silly in retrospect, but at the time we were absolutely terrified of this. So the government of Canada and the United States decided to send a lot of personnel to the north to make sure that this didn't happen. There were a number of northern military bases established, and the distant early warning stations were set up. So there was a lot of military, a lot of scientists, a lot of engineers, and other associated personnel that went up north. And when they were there, they came across the North's most abundant natural resource, biting flies. And the sheer abundance and annoyance from these flies pestered the governments of both countries into learning more about the northern fauna. So one of the ways they went about this was by developing the Northern Insect Survey, which started in 1946 and ended in 1961. And it ended up contributing 750,000 specimens to the CNC. It was an immense undertaking and included 66 expeditions to 70 locations. And a lot of entomologists were hired, and they were kept very busy. They were flown up north by the Canadian military. They were given these cool little tank things, center right, to drive around in across the tundra. And towards the top left there, you can see in their free time, they were actually asked to test pesticides. So those are entomologists that have rolled up their sleeves and their pant legs, and they have different pesticides on, and they're counting and collecting the things that are biting them. Very dedicated people. Another survey that was initiated was the Forest Insect Disease Survey. And this was developed by the Canadian Forestry Service, again, from the 40s. And it employed a lot of people that collected about 2 million specimens. And half of these, 1 million, are deposited in the CNC, because the CNC was where the material was sent to either be identified or have identifications confirmed. And half of the material was retained as vouchers, and the other half was sent back and is now located in various collections across Canada, mostly in Ontario. Before the war period, however, there were a number of other expeditions to the north and the west of the continent, and it was also very nationalistic in nature. It was not necessarily about learning for learning's sake. Canada was developing at the time, the borders were not set, and part of the way that they established territorial rights was by having a person there recording stuff and taking things to say that we were there first. This is ours. So they were basically scent marking the land. So this is evidenced in things like the Geological Survey. Joseph Berterel actually collected a lot of insects and arachnids like mites. There's a great biography about him that I recommend that you read. There's the Canadian Arctic Expedition and the Eastern Arctic Patrol. And to really reinforce this nationalistic aspect of the collections, the main point of the Eastern Arctic Patrol was, as evidenced in these photos I found, to send Mounties to a lot of locations along the coast up to the Arctic, where they would erect the flag, salute it, and then go on to the next station that they decided was important for territorial sake. So it was really about scent marking Canada to say this is ours. In the post-war period, there were also a number of international expeditions because the scientists in Canada were really capitalizing on the knowledge that they'd been accumulating, and to get a good understanding of the local fauna and to understand these taxa they were studying that they had in Canada, they realized they had to have an international context. A broader view was needed to learn more about these things. So there were many expeditions to the Neotropical, Nearctic, Palearctic, and Oriental regions, two of which were expeditions to Nepal and Mexico, because researchers at the CNC wanted to look at the differences between in the transition from the Nearctic to the Neotropical and from the Palearctic to the Oriental regions. So they went to the transitional areas. There were also a number of individual collecting trips by separate researchers supporting their own narrower taxonomic interests. And the collection also continued to grow through donations and collection amalgamations and other things. Now, moving on to digitization, because that's what we're all interested in now, isn't it? Please, I urge you to write down the address of our online portal to our database. We're a government organization, and unfortunately, our database is now deeply buried under pages and pages of government bureaucratic websites. So you can't just Google the website anymore, unfortunately. So we've now made the transition to making all specimens being born digital. So every time we put a new label on a new acquisition, it has the unique identifier on it that links it to a database record. You can go into our database and search for all digitized specimens through our search page. You can go to individual specimen records, and if there's images, you can click on those for a large resolution image. Right now, we've only posted 5 megabyte JPEGs, but if you request the original 35 megabyte TIFFs, you can feel free to use them for your own research purposes. So please get in touch with us if there's any images on the database that you want to use or data. As of last January, we've databased over 2.5 million specimens, which is about 14% of the collection. And we focused largely on making sure that we got full coverage of our name-bearing types. So all of our types are fully digitized. And with regards to images, we've imaged all of our pinned types and half of our slide-mounted types. So the remaining specimens to be imaged are fluid-mounted and about half of our mites. So once that's done, we'll have images for all the specimens in our collection. Now, we were lucky enough to receive funding over the last 6 years from the federal government to digitize our collection, or as much of it as we could, in order to make this data more accessible, to release the data, because we all know how hard it is to access that knowledge, even for those of us who are sitting right beside these cabinets. So we received funding to set up some infrastructure, and we found it very beneficial to use most of our money to invest in people. So we trained students and technicians very thoroughly to make sure that we captured good, high-quality data and that it got well vetted, it was checked, mistakes were corrected. So we were releasing research quality data. One individual we found could digitize 200 to 400 specimen records a day, but towards the end it was closer to 200 because we found it made a lot of sense to image our labels as part of our digitization pipeline. You could see some example labels there on the screen. And this was extremely useful because it made sure that transcription was more efficient, there were far fewer mistakes, and it was easier to locate and correct mistakes without having to run back and forth to the collection. And we all know how time-consuming that be, especially if the name on the record was incorrect. So it's really hard to find a specimen if the wrong name has been added to the specimen record. So this saved us a lot of stress and increased the quality of our data quite a lot. So the stations you can see there were relatively affordable, so it only cost about 6K Canadian. And these were actually also used for our specimen imaging. It was very affordable to get an additional lens, a macro rail, some stacking software, and this produced very high quality research grade images for specimens down to about 1.5 millimeters, which is really great because we basically invented this system ourselves, especially with the input from Henri Goulet in the Hymenoptera unit. And to get specimens that were smaller than that, we of course needed to get separate, more expensive equipment. But now we've come to almost full coverage of our name-bearing types through these setups. So that's it, Shell. Thank you very much for listening to my talk. And please, everyone, appreciate the role of biting flies in the development of all of our collections. Thank you very much.
So next up, I'm actually going to welcome someone to the stage to present more of a traditional style conference talk. So the next tour is of the Museo de Arthropodas de Baja California in Mexico, and we'll welcome William Clarke.
So my name's Bill Clarke, as Nicole said. Sarah's in Mexico today. Our objective is to introduce our objective is to introduce the collection at Secesi to you. Thanks to Nicole and Jennifer and Andrew for making this section happen. And probably thanks to Luc LeBlanc for originally suggesting that I give the presentation. For a quick geography lesson, Ensenada is where the Secesi collection is located. It's about an hour and a half south of San Diego. The campus is large, covers all of the sciences, and it is right across Highway 1 from the University of Baja California. So it's right on the north side of town, so it's easy to find, easy to get to. The collection is currently, we moved it recently, and it is in the top of the biomedical building. It's in the Department of Conservation Biology. The building now has 4 floors on it, but it's located in that building. We even made the building directory, so feel pretty good about that. Myself and Sarah are I guess the collection managers for the collection. Sarah is more the go-to person to contact as she is physically there. But I also assist with loans and donations of specimens and such. Kind of bring back some old ones. I scared a couple of people today, told them I'd have a photo of them in here. This was the good group from the ECN ESA meetings in 2007. You'll recognize some faces there. So we've been trying to get the word out for a while on the collection. This is in front of the old building. Introduction, objectives. An arthropod insect collection was needed in Baja California. There is a small collection in the south in La Paz at Sibnor, primarily dedicated to spiders and some general collections. The Secesi collection is affiliated with our museum in Idaho. We established the collection in 1997 and have used it for vouchering various biodiversity projects. It's also receives a fair amount of educational use with mostly with master's thesis students and undergraduates presently. We do have a pretty good outreach program, lots of, for Northern Baja California, lots of different programs out to the public. So what we have is the collection room. It's in conservation biology again, dry collection of pinned and pointed materials, ethanol materials, small library, kind of the normal equipment you would have with such. Something a little bit unique to the Mexican system is the ability to have undergraduate students assist in community service and professional service. So it's essentially kind of free labor for those that they pay back part of the cost of their education. We also bring some of the students to Idaho, so it's a back and forth process. The facilities for the pin collection, it's primarily California Academy of Science system. We have 18 cabinets, roughly 424 drawers, so it's a small collection. The collection has increased quite a bit in the last 4 or 5 years due to primarily acquisition of student projects, graduate student projects, and undergraduate student projects. And roughly 80,000, probably more than 80,000 pin specimens at this time. The primary emphasis is in the holdings are Coleoptera, Hymenoptera, and Diptera, but most all groups are represented. Alcohol collection is similar to alcohol collections we've talked about today. It's small, probably primarily arachnids and scorpions. A few results to date. Lots of new species, lots of new records. Have the curators and associates have been involved in various biodiversity surveys around the peninsula. Some of these surveys have occurred in these areas marked. Mexico has a lot of protected areas in Baja California. All of the colored areas are some sort of protected area. We've done surveys in the very southern spot there in the Sierra de la Lagunas. We've done surveys along the Gulf Coast around Loreto, and then in that sort of that brown central desert areas where predominant amount of our work has taken place. These protected areas are different from probably what you expect in Canada and the US and maybe other places. They're protected areas. The enforcement is lacking just because of personnel. What we can really use from this group is visiting entomologists. Contact Sarah if you would like to visit. Help work on the collection, and we can also use Baja collected specimens from over the years. And please contact me if you have anything like that you would like to contribute. I can hand carry those down this winter. We are affiliated with major Mexican institutions, governmental institutions, and with our museum again in Idaho, and obviously with students at OABC and SECESI in Ensenada. And that's what I have. Any questions, if there's time?
How would you get loans in and out? That you take them every year, or you mail them?
I would not mail them. Right now that's done either between agreements with San Diego Natural History Museum in San Diego back and forth, or I spend a third to half the year in Baja California each year and can easily transport materials. Hand carry is probably the safest.
Thank you very much.
Thank you.
Yeah, we can have it. So this is the last talk prior to the break, and even though we are 5 minutes in front of the schedule, I believe that most people are going to be online already, so we are going to start it very soon. So this is a tour of the collection of insects of the University of Costa Rica by Humberto Lezama. This presentation, it doesn't have any sound on it, so it's not an audio issue with it. And I also want to shout out that this is the first collections tour in the entire 3 years of running this from Costa Rica. So it's really great to be able to see some different collections around the world. So Humberto is joining us online and is available for questions. So if anyone has any questions in the room, I can repeat them into the microphone for him to hear.
Can you hear me?
Yes, we can hear you.
Okay, I will be more than happy to answer some questions.
Yeah.
Video. So many awesome cool effects out there. I was wondering if you actively like spread the antennae of all of your eagles because they were quite delicate.
So the question is firstly a compliment to the video, but it was about whether or not the weevils in the collection, if there is effort to spread the antennae, because they were all very beautiful in the images.
Yes, we used to do the spread out the antenna because it has a very nice taxonomic characteristics. And right now we are doing a strong effort to do a good mounting process at the collection, and the new specimens has a beautiful mounting, and also we have very nice photographs of some of them that looks like a painting.
Perfect. And then we have a question over here. Tommy?
How many specimens are there?
The question is about approximately how many specimens are there in the collection?
Around, this is a small collection, but well represented, you know. But we estimated around 3,000, 300,000 different specimens, mostly in Coleoptera. It's a well represented collection in according with a huge biodiversity.
Is there any other questions in the room or online? You have one more question over here?
Yeah, I noticed cerambycids were featured prominently in the video. Can you tell me more about the cerambycid?
So the question is about the cerambycid holdings within the collection. Would you be able to tell us a little bit more about them?
Well, this is a really good collection because I was collecting with strong people like Frank Hobor, Ed Gisbert, and Ubirajara Martins in Brazil. And it's a huge collection according to our standards. And it's well mounted because I spent a long time collecting longhorn beetles around it. And then I have also a lot of nice photographs. And it's if somebody wants to see the collection of longhorn beetles, could visit us and will be more than welcome. And we have a very nice staff around it.
Excellent. Thank you very much. Actually, I have one more question. In the video, you were spraying down the labels with the varnish, and I think a lot of I've never seen that before. Does that help with the longevity of the labels, or is there a reason in particular you've chosen to do that? And how long have you been using this technique?
Okay, for long. Because
and what do you use?
Okay, well, it's quite interesting because remember, we are in that really tropical country and rains a lot and there is a lot of humidity. And therefore, also our labels, they are printed on what is a laser printed. If you apply a coating of this art varnish, you are preserving the label and avoid to mold growing or dust or something like that. Literally, the label is impermeabilized, you know. And also it's quite interesting because for example, when in my country, when it is raining, we have outside 100% relative humidity. And therefore, we try to preserve the label because it's quite difficult. And then we use art varnish special for preservation because also it has UV filter. And also, you know, we're preserving the label for really longer.
Thank you very much. That was fascinating. And really, we appreciate you joining in and sharing the collection.